Hello, good afternoon participants. Welcome to the webinar on selection of appropriate tests of statistical significance for health science researchers. I am Dr. Sandeep Sugadan, Associate Professor in Community Medicine, Quest International University, Malaysia. We have divided this webinar into two parts. First part is understanding the terms. It is actually basics in inferential statistics. Second part is selection of appropriate tests of statistical significance. I will give you an introduction about this webinar. As a health science researcher or academician or a medical researcher or a student in health science, we have to do a lot of research. We will be collecting data and we will be analyzing the data before it is presented in a conference or published in an international journal or an index journal. So the knowledge and skills to select appropriate test of statistical significance is very important for us. We need to select which test to be selected in which scenario and how to interpret the results of that test. So we will start our journey. First part, we will understand some terms. Learning outcome of this first session is, at the end of this session, the participants should be able to describe the following terms. What is inferential statistic? What is dependent variable, independent variable, null hypothesis, alternate hypothesis? Alternate hypothesis is the other name for research hypothesis. P-value and errors in research such as alpha error, beta error and power of study. So we will go through each terms. Inferential statistics. It is otherwise known as statistical inference. It is a process of applying statistical methods in order to draw conclusion or in order to reach an inference about population from the sets of data which is collected from a sample. We are collecting data from a sample and we need to generalize it to the population or to the reference population from where sample is drawn. The reference population is otherwise known as sampling frame from which we are drawing the sample. Inferential statistics is the process of applying statistical method to come to conclusion about population from the data which is collected from a sample. It is otherwise known as analytical statistics. Now we can know what inferential statistics does. Inferential statistics makes inferences about population, about data drawn from sample from that population. Okay, already we explained that. Inference or prediction of association between a dependent and independent variable. Okay. We are making an inference or prediction that if there is an association between a dependent and independent variable. Third one, prediction of difference in dependent variable between two or more groups of independent variable. So we are predicting the difference if there is a significant difference in dependent variable between two groups or more than two groups. If there is a significant association between a dependent variable and independent variable. These are the main things we are getting from the inferential statistic. Now we can see what is a variable. Variable is an element, feature or factor that is liable to change or vary. Whatever vary is a variable. Variables in a study can be classified into dependent variable or independent variable. Now we can see what is dependent variable, what is independent variable. Dependent variable, it is otherwise known as outcome variable. It is a factor or phenomenon that is changed by the effect of an associated factor called the independent variable. Dependent variable changes based on the change or manipulation of independent variable. So, why it is known as dependent variable? Because it is dependent on value of independent variable. Okay. It is otherwise known as outcome variable also. What is independent variable then? Independent variable, it's otherwise known as predictor variable. Okay, independent variable is typically the variable which is manipulated or changed. And the dependent variable is the variable which is affected by the manipulation or changes in the independent variable. Now we can see an example, then you will understand better. Regular physical activity can affect the variable body mass index. So in this scenario, body mass index is the dependent variable because body mass index is dependent on regular physical activity. And a regular physical activity is the independent variable. Why? If you are changing or manipulating regular physical activity, you can change the dependent variable, that is body mass index. Obesity can lead to development of type 2 diabetes mellitus. So here type 2 diabetes mellitus is the dependent variable because it is dependent on obesity. So obesity is the independent variable. Okay. If you manipulate or change obesity, that is independent variable, you can change the dependent variable, that is development of type 2 diabetes mellitus. So we can understand same variable can be either dependent or independent based on this scenario or situation. Inferential statistics checks if there is a real difference in dependent variable in two or more groups. That means there is a statistically significant difference. I will give an example. Hemoglobin values among males and females. So we are taking hemoglobin values of males and hemoglobin values of females, and we are calculating the mean value of hemoglobin among males, mean value of hemoglobin among females, and we can apply inferential statistics to check if the difference in mean hemoglobin value among male and female is it significantly different, or if the difference is statistically significant, or the difference is real. Okay, another example. The same hemoglobin value we are testing in 3 different groups like Malay, Chinese and Indian. These are the 3 ethnic groups in Malaysia. So we are checking hemoglobin values among Malay, hemoglobin values among Chinese, hemoglobin values among Indians. And we are checking if difference is statistically significant, or there is a statistically significant difference. We are applying inferential statistics to check that. Another one, if there is a real association between a dependent and independent variable, that means there is statistically significant association. I will tell you one example. Smoking is leading to lung disease. So dependent variable here will be lung disease and the independent variable will be smoking. We can apply inferential statistics to know if there is any association between smoking and lung disease. If there is any statistically significant association between smoking and lung disease. So if there is a higher proportion of smokers are getting lung disease compared to proportion of non-smokers getting lung disease. So that means there is a difference or significant difference between smokers and non-smokers related to dependent variable. Okay. Test of statistical significance are applied to know if the difference between mean values in two or more groups is significant. Difference between mean values in two or more groups is significant. Or the difference between proportions in two or more groups, males and females. Proportion of diabetes among males, proportion of diabetes among females. Is it significantly different? Or the difference in occurrence of disease or a problem between two or more categories. Among smokers, what is the occurrence of lung disease? Among non-smokers, what is the occurrence of lung disease? If the difference is statistically significant. Test of statistical significance are applied to know if the difference is statistically significant in these three types of scenarios. Before applying appropriate statistical tests, the researcher need to have a null hypothesis. You need to understand what is null hypothesis. Okay. Null hypothesis is a general assumption that there is no relationship or association between dependent and independent variables. Okay. Or another assumption is that there is no difference between dependent variable in different groups of independent variable. Okay, null hypothesis is a general assumption that there is no relationship or no association between dependent and independent variable, or no difference between dependent variable in different subgroups. If there is any difference or association seen, it is due to chance alone. This statement is known as null hypothesis. Alternative hypothesis or research hypothesis. Okay, it's a general statement or assumption that there is a relationship or association between dependent and independent variables, or difference between dependent variable in different groups. Statistical test of significance is applied to prove or disprove the null hypothesis. Okay, statistical test, either parametric test or non-parametric test is applied. We need to check if the difference or association is statistically significant or not. If statistically significant difference, we are disproving the null hypothesis. If the finding is not statistically significant, we are proving the null hypothesis. Inferential statistics is done by using parametric or non-parametric tests of statistical significance. Then look for the significance value or p-value. We need to look for significance value or p-value. After that, okay, what is p-value? P-value or significance value is the probability to have an association between the dependent variable and independent variable due to chance alone. Probability to have an association between dependent and independent variable due to chance alone. Or in another terms, we can tell probability to have a difference in different subgroups due to chance alone. That is p-value. If p-value or significance value in the parametric or non-parametric test is less than 5% or less than 0.05, then the difference or association is real and not due to chance. Okay, so I will repeat once again. P-value is the probability to get the association or difference due to chance alone. So if p-value is very high means it is due to chance. P-value is very low means it is not due to chance. P-value below 5% or less than 0.05, that means that is a cutoff for not due to chance. Or we can say if p-value is less than 0.05, the association or difference is real. Okay. In other term, association or difference is statistically significant and it is not due to chance. Okay, understood? Similarly, if p-value or significance value in the parametric or non-parametric test is more than 5% or more than 0.05, what it means? P-value means probability to get the association or difference due to chance. If it is high means it is due to chance. If it is more than 5% means it is due to chance. The difference is due to chance. Association is due to chance. Association or difference is not real. So it is not statistically significant. Association or difference is considered not real and due to chance alone. So not statistically significant. Errors in a study. If you are doing a study, it can have different possibilities are there. Okay. First possibility is that null hypothesis is really true in the population. Okay. Null hypothesis is really true in the population, and in your study also you are accepting null hypothesis. So null hypothesis is really true, and in your study also you are accepting null hypothesis. That type of studies are considered as correct study. Null hypothesis is true in the population, and in your study you are accepting null hypothesis. Another possibility, null hypothesis is really true, but in your study you are finding that null hypothesis is not true and you are rejecting null hypothesis in your study. So what is a possibility? Null hypothesis is really true in the population, but in your study you are rejecting null hypothesis. That is known as type 1 error or alpha error. Understood? Then third possibility is null hypothesis is really false in the population. Null hypothesis is really false in the population and you are rejecting null hypothesis. In your study also you found that null hypothesis is false and you are rejecting null hypothesis. Okay, what is the possibility? Null hypothesis is really false and you are rejecting null hypothesis. That is correct, right? That is known as correct study. So in that case, power of study is also good. Okay. Another possibility. Null hypothesis is really false in the population, but in your study you made a mistake. You are accepting the null hypothesis. Null hypothesis is really false, but you are accepting the null hypothesis. That is known as type 2 error or beta error. Understood? Type 2 error or beta error. Okay. Type 1 error or alpha error. I will give an example. Okay. What is that? From the previous figure, type 1 error means null hypothesis is actually true and you are rejecting the null hypothesis, right? Null hypothesis is actually true and you are rejecting null hypothesis. I will give an example of null hypothesis actually true. Association between height and intelligence. Okay. What is the null hypothesis? There is no association between height and intelligence. So null hypothesis is actually true in the population. Okay. But if in your study you are getting that there is an association between height and intelligence. So if null hypothesis get rejected in the study you got an association between height and intelligence and you are rejecting the null hypothesis in your study. Okay, that is known as type 1 error or alpha error. Is it clear? Now, the probability of rejecting the null hypothesis when it is actually true. Okay, the probability of rejecting the null hypothesis when it is actually true should be less than 5% or less than 0.05. That means that alpha error or type 1 error should be less than 0.05. Now we can see the example of type 2 error or beta error. Please remember the diagram we saw before. Type 2 error means null hypothesis is actually false in the population. But you are accepting the null hypothesis in your study. I will give an example where null hypothesis can be actually false. Association between smoking and lung disease. What is the null hypothesis in this? There is no association between smoking and lung disease, right? But that null hypothesis is actually false in the population. Null hypothesis is actually false in the population. But in your study, you are getting that null hypothesis is true. If in your study you are telling that there is no association between smoking and lung disease. So if null hypothesis fail to get rejected or if null hypothesis get accepted in the study, it is known as type 2 error or beta error. Okay. The probability of failing to reject null hypothesis. Okay. Probability of failing to reject null hypothesis or probability of accepting null hypothesis when it is false should be less than 20%. So beta error, that is beta error, right? Probability of failing to reject null hypothesis when it is false is known as beta error. It should be less than 20% or less than 0.2. That means power of study should be 80% or more. Power of study means it is 1 minus beta. If beta error is 0.2 or 20%, we have to calculate as 20 divided by 100, that is 0.2. If beta error is 0.2, 1 minus beta will be 0.8, right? Power of study should be 0.8 or above. If we are taking 20 as beta error, 100 minus 20 will be the power. Okay, 20% is the beta error, 100 minus 20, 80% will be the power of the study. So we should have good power. that is more than 80% or less than 20% of beta error in this study. Okay, now we are finishing the first part of our webinar. Thank you so much. We can move on to second session.